Breaking the wall to mRNA vaccines. How a novel approach helps combat the COVID-19 pandemic. Özlem Türeci, BioNTech. On November the 9th, 1989, I was following the fall of the Berlin Wall from my tiny student apartment in Hamburg.
Thank you very much for this warm welcome. Thank you, Sebastian, Turner, and the Falling Walls Summit team for this invitation. Thank you, Ministerin Karliczek, for this words of wisdom. It's my pleasure to be here today at the Falling Walls Summit in this session, which is about the major breakthroughs of the past year. The wall we helped to tear down was an invisible one. It was a virus which separated families, countries, and continents. Luckily, what we had up our sleeves was something we had been working on since our very early days in academia. A deep knowledge of the human immune system and messenger RNA technology. Building on our mRNA technology, we were able to do the seemingly impossible, to develop a vaccine against a newly emerged virus within less than 1 year. Project Lightspeed was the fastest development in the history of medicine, fast, but according to high standards, both scientific as well as ethical. BNT162b2, aka Comirnaty, became the first approved mRNA drug. A first drug based on mRNA in the history of medicine, an example that science and technology can lead to breakthroughs. But actually what happened in the last year, what happened in 2020, is not what I want to talk about. Today I want to talk about the decade-long journey of basic research, of development efforts that prepared us for this. Yes, breaking down walls takes years of preparation. Isn't that what we all have learned from history, the hard way, over and over again? And what prepared us was a journey that started in the early 1990s. The original mission back then was a different one, as was the enemy we wanted to defeat, cancer, against which we wanted to develop better medicines for our patients. An ideal cancer treatment, we thought, must harness the human immune system. After all, this is the only system which is capable of relentless force, of a predator's adaptability, and of high precision, if properly instructed by a vaccine with ideal features. An ideal cancer vaccine, we thought, must be of tremendous potency. At the time it is detected, cancer has already invaded the body with billions of cancer cells. And it becomes a numbers game to defeat this disease. A numbers game which needs the deployment of a large army of immune systems forces. And ideally, we thought, a cancer vaccine must be truly individualized. The root cause why current cancer treatments fail is that every patient's tumor is unique. Why then shouldn't we design vaccines based on the genetic profile of the unique patient's tumor and customize on demand a vaccine for each and every patient? Yes, and this then became our grand mission, to make a difference for the individual cancer patient, for the single human, by harnessing the immune system. In the decades that followed, we became wanderers between different worlds. Scientists, physicians, innovators by training, but also entrepreneurs and drug developers, because the mission called for it. And we created our own humble microenvironment, a special ecosystem which would allow us to translate our science to something more meaningful, to survival. In those decades, we studied the human immune system on the level of basic science in all its depth and in all its beauty. And in those decades, we embraced various technologies, and in that process realized that mRNA, that messenger RNA, could be an ideal cancer vaccine platform. mRNA, the most primal information technology, created by nature and present in all of the cells of our body. We realized that it had potential, but we also understood that improvements would be needed, technological and conceptual ones. The problem of mRNA back then was that it was of low potency, which meant that even high doses of mRNA had little effect, had little immune response. This was the reason why back in the late 1990s, not many believed in the potential of mRNA as a drug class. In the years that came, we spent a lot of efforts to optimize mRNA and to make it suitable for the purpose of a usable drug in humans. And we were not alone in this. The advances of many scientists in adjacent fields contributed to this and helped us to make mRNA better as a drug platform. And as any breakthrough, it is the input, it is the many efforts of scientists, of generations of scientists, that made the practical use of mRNA feasible as a drug platform. Let me show you on the next slides. On these slides, three of our many improvements, of our key or core improvements, which we made in order to make mRNA a usable drug class. The first innovation is about molecular engineering, increasing mRNA potency by design. The goal of a vaccine is to make the immune system aware of the enemy to attack. mRNA does this by delivering the blueprint of the wanted poster, of a protein, which is the key feature of the enemy. And this was, back then, a very inefficient process. However, by nature, mRNA not only delivers this blueprint of a wanted poster for the body to produce, it also comes with instructions, several and diverse instructions which tell the body how to produce. We spent many years to research each of these instruction elements and to design each single of them to improve the potency of mRNA. And in combination, we achieved a much higher yield of protein, a much higher yield of the wanted poster, which resulted in more than 1,000-fold increase of the ability of mRNA to trigger an immune response. And in parallel, in the US, meanwhile, our colleagues Katika Rico and Drew Weissman discovered an additional way to contribute to the higher protein yield, namely by substituting one of the letters of the 4-letter code building block of mRNA. Innovation number 1, the body hack, bringing mRNA to the right cells at the right places. The immune system has specialized cells, so-called dendritic cells, which act as generals, as high-performance trainers. And a vaccine should ideally bring the wanted poster to these cells, which then activate in the most efficient way the different immune forces which are gathered around of them against this enemy. But not all dendritic cells are created equally. The most capable among them reside in special organs, in so-called lymphoid organs, which is the body's network of immune system boot camps. And ideally, a vaccine would bring the message to exactly these cells in exactly these organs. We developed technologies, lipid-based envelopes, which did this job, which targeted our mRNA message to the right cells body-wide in order to enable as many generals as possible in as many boot camps as possible to train the different immune forces of the immune system. And innovation number 3, which we developed in order to have better individualized cancer vaccines, is about rapid manufacturing on demand. This process of identifying the patient's cancer profile on a genetic level, of manufacturing a vaccine of unique composition so that it is ready for administration for each and every patient has been a race against that patient's tumor. We have produced hundreds of such customized vaccines since 2012 in our clinical trials for patients worldwide to be vaccinated against their cancer. And each of these productions meant running the race over and over again. When we started this in 2012, 2014, it took us 3 to 5 months of handcrafted production for each and every patient to deliver their vaccine. In 2019, shortly prior to the pandemic, we had this down to 3 to 4 weeks by robotics and automation. So at that fateful last weekend in January 2020, when we realized that humankind was actually already in the midst of a pandemic, that the world as we knew it was in danger and people we loved and cared for were at risk, we were prepared because of those years, of those decades of perseverance and grit. We had full command over our RNA technology. It was routine for us to react. It was routine for us to go fast through this process of starting with genetic information, designing a vaccine, and manufacturing it. Our team was well trained and accustomed to solving problems in real time, and we had optimized for potency. What this means, I will show you in a minute, and I have help here. Optimizing, thank you, for potency means that it takes little to achieve a big and significant biological effect. And this optimization, why this bag of underwhelming size holds more than 1.6 doses of our mRNA vaccine, of the COVID-19 vaccine BNT162b2. And this means why we were able to deliver for the entire humankind. Thank you. So this all meant that we were prepared for delivering the vaccine which was needed, and we should learn from this. Let us not wait for the next big problem, the next crisis of unprecedented scale, to embrace technologies which are powerful, to embrace scientific concepts which may help us even today to react to the problems which have global scale. We can tear down walls. Let us do this together. Thank you.